The focus of my laboratory over the last 20 to 25 years has been trying to understand one of the most well-characterized interventions that actually increases not only lifespan, but health span. And this is an original observation by Clive McKay showing that calorie restriction of mice or rat can actually really dramatically increase lifespan, as shown on this first slide. And our model system has been that calorie restriction and its effect on health span and lifespan is not only increasing lifespan, but also increasing health span, as shown here. And you can see the chronic diseases of aging that we are all familiar with, I think, after which this meeting is named, has really been equally modified by calorie restriction. So the question is, what is in this black box? And we have been focusing on a series of metabolic networks, critical metabolites such as NAD, acetyl-CoA, S-adenosyl methionine, alpha-ketoglutarate, or beta-hydroxybutyrate, that we know fluctuate as a function of calorie input. And what we have been trying to understand is how variation in the levels of these metabolites is able to affect the activity of a number of critical epigenetic regulators. And I've listed them as pairs. The NAD is known to regulate the activity of sirtuins, acetyl-CoA, the activity of acetyltransferases, and so on. Today, I'm going to be focusing on one of these pairs, which is the NAD-Sirtuin interaction. We've heard a lot about epigenetic reprogramming yesterday, and to put this work in the context of this other work, it's good to realize that many of the proteins that I'm showing you in the yellow bar are actually epigenetic regulators. And one thing that we have not discussed very much is why is there an epigenetic drift during aging? Our hypothesis is that degradation in these signaling networks between the metabolites and the epigenetic regulators is at least in part responsible for the epigenetic drift. So the thinking is that rather than reprogramming, which obviously is an exciting approach that will be further pursued, we also would like to prevent the epigenetic drift at the first place. And this is the focus of our work. So talking about the sirtuins are named after a protein called Sir2, which is an NAD-dependent protein deacetylase. It is illustrated here in the middle. The Sir2 protein, originally isolated in yeast, is able to remove an acetyl group on the left from histone proteins, but it does this in a really unique manner by coupling the deacetylation to a breakage of NAD, which is shown at the bottom. Here is NAD, and generating two different products, ADP ribose, which serves as an acetyl acceptor. So the acetyl from the histone is transferred to this ADP ribose, and nicotinamide. So in essence, this protein is able to sense the amount of NAD in its environment and to regulate its activity, its deacetylase activity on histone as a function of NAD concentrations. Now, Sir2 is part of it is part of the calorie restriction response, and this has been documented in multiple organisms. What I'm showing you here is yeast, C. elegans, Drosophila, and mammals all have various sirtuins. These sirtuins enzymes, or these proteins, if removed genetically from the organism, abrogates the calorie restriction response. And this has been illustrated in many of these organisms, including in mammals. Now, in our work started in this area by cloning the human sirtuins. A student in the lab, Brian North at the time, essentially searched the human genome for orthologs of Sir2 protein, was able to identify 7 proteins that we cloned. They share a catalytic domain, which is the deacetylase domain highlighted here in blue. But it was striking also that they had variable N and C-terminal extensions, which we have shown since contribute to different subcellular protein expression and different activities. And most important thing to know is that 3 of these proteins 31, 36, 37, and perhaps also 35 are localized in the nucleus partially and can be functioning as epigenetic regulators. 33, 34, and 35 are predominantly mitochondrial protein, and we spend a lot of time working on those. And finally, 32 is predominantly a cytoplasmic protein. Now, let me review a bit what we know about NAD metabolism. The first thing to know is that NAD metabolism, so the cofactor for the Sirtuin, is actually differentially regulated in different cellular compartments. And I won't go through all of the regulation of the pathways. We will go through this a little bit later. What is important to know is that NAD levels in different compartments vary and therefore regulate the activity of the Sirtuins and other proteins that depend on NAD for their activity. For example, I'm showing you here the mitochondria where NAD is affecting the activity of 33, 4, and 5, or in the nucleus, for example, where NAD levels are affecting 31, 36, and 37, but other proteins such as PARPs, CD38, SARMs, and so on. So we have actually focused in my lab primarily on 31 and 33 as two enzymes, and the analysis of their targets using mass spectrometry to identify proteins whose acetylation or acylation level is regulated by these different Sirtuins, identified a number of key regulators of aging. And I just pointed out a few here. I want to point out NF-kappa B, for example, which is a critical factor, master regulator of the inflammatory pathway, is regulated by 31. FOXO1, FOXO3, key enzyme, key transcription factors regulating aging, also in response to oxidative stress, are also regulated. PGC1 alpha, a master regulator of mitochondrial biogenesis, and the list goes on and on. And based on the identification of these factors and their regulation by Sirtuins, it is not a stretch to imagine that the Sirtuins play a really key role of master regulators of these enzymes. Now, one of the things that was remarkable is the discovery by many groups that the activity of 31 and 33 and other Sirtuins decrease during aging. And therefore, a model was proposed that one of the causative events of aging might be this downregulation of Sirtuins. Now, the reason why their activity decreases because NAD levels themselves have been shown to decrease during aging. And this has been actually demonstrated in multiple organisms. This is a review by Yoshino and colleagues in Cell Metabolism in 2018, really putting together all of the evidence that NAD levels decrease during aging. This happens in multiple organs, including the brain, the liver, adipose tissue, pancreas, and so on. It also happens in multiple organisms. This has been documented in some tissues in humans, in mice, in rats, and so on. So this, we believe, could be one of the really important drivers of the epigenetic dysregulation that we know occurs during aging. So let's try to, as a next step, try to review why could NAD levels decrease during aging. And this is an important slide because it will review essentially much of the work that I will be discussing afterwards. So there are two biosynthetic pathways that lead to the synthesis of NAD. One is the kynurenine pathway, which uses tryptophan and a series of steps to go to NAD. NAD is right here. The second is so-called Price-Handler pathway, which uses niacin, which is nicotinamide or nicotinic acid, the two different forms, which can be used as a vitamin, can be used as a precursor, also leading to the synthesis of NAD. Now, both pathways are obviously important. If there's a niacin deficiency with low level of tryptophan, this leads to a disease called pellagra, which can actually be mortal. Right now, all our food is supplemented with niacin, so there's no evidence, at least in the US, of deficiency in these precursor vitamins, either tryptophan or niacin. So it is generally considered that the defect that we see in aging is not caused by a biosynthetic defect. The second important thing to know about NAD is that there are two different forms of enzymes that use NAD. The first family of enzymes are dehydrogenases that use the redox couple between NAD and NADH to operate a series of redox reactions. And these, in many actually intermediate metabolic enzymes in the Krebs pathway, in glycolysis, and many other pathways actually use NAD, the NADH couple. There's also a separate family of proteins that we call the NAD-consuming enzyme, and I'll be spending a lot of my time talking about them, that do not only use the NAD form, do not use the NADH form, which by the way is actually in large majority, only a small fraction of the NAD is in the reduced form, NADH. And so these NAD-consuming enzymes, just like the Sirtuins I mentioned, take NAD and generate nicotinamide. And this nicotinamide has then to be recycled in two steps, one driven by an enzyme called NAMPT, the other one driven by three different enzymes called NMNAT 1 and 3, which are present in the nucleus, in the cytoplasm, and in mitochondria, and therefore lead back to the synthesis of NAD. Now, why is this salvage pathway important? Because we know that there is enormous flux through the system. If we inhibit, for example, the enzyme NAMPT with a small molecule inhibitor, within a few hours, NAD levels come down to close to zero and the cell die. So this NAMPT plays and NMNATs play a critical role in recycling the nicotinamide into NAD. Now, why is this important? Because given the amount of flux that happens through the system, we suspected that this might be where the problem occurs during aging. And so the hypothesis that is emerging is that if you think about the Sir2Is and the PARPs, all using NAD, consuming NAD during their activity, one could imagine that under conditions of aging, there's increased DNA damage, for example, and this has been documented. That could lead to an overactivation of the PARPs, and therefore to a siphoning of the NAD towards the PARPs. Now, a really good example of this, almost caricatural example of this, are a series of diseases in which there's DNA damage repair problems, such as cocaine syndrome or xeroderma pigmentosum or ataxia telangiectasia. These are three diseases in which there are defects in DNA repair. Those have chronic DNA damage, even early in childhood, and chronic PARP activation. And it's been known that in every one of these diseases, there are dramatically lower NAD levels, and one can actually restore some of the pathological defect by providing NAD precursors, as I will be showing later. Now, we proposed, Carlos Canto and myself and Johan Ulrichs proposed a hypothesis that a siphoning of NAD towards other NAD-consuming enzyme might be responsible for the decrease in NAD available for the Sirtuins and therefore to some of the manifestations of aging. Today, I'll be speaking about another enzyme that we think also plays a role in the siphoning. This is primarily CD38. There's another enzyme called CD157, which is similar to CD38, but does not appear to be playing the same role. So what is CD38? So CD38 has actually been known for a long time. It's an NAD metabolizing ectoenzyme. It is present at the surface of cells. In most cells, the majority, but not all of CD38 is facing outward. In macrophages, for example, close to 30% of CD38 actually is facing inward. It is found on immune cells, CD4, CD8 T cells, B cells, and NK cells. It's been shown to function in cell adhesion, in signal transduction, in calcium signaling. And interestingly, it's highly expressed in multiple myelomas, where it has actually been used as a therapeutic target to treat multiple myeloma. This is a drug called Darzalex. And so why do we become interested in CD38? This was based on a paper published by Eduardo Cini and his colleagues at Mayo Clinic, where they showed that CD38 levels actually increase during aging. And this is illustrated here on a Western blot. You can see CD38 expression going up. SIRT1 not changing, and AMPT not changing. Again, this is the key enzyme in the salvage pathway. And the consequence of this is that when you look at NAD levels in adipose tissue, you can see them progressively decreasing as a function of aging. But remarkably, what drew our attention in this paper was additional data looking at a CD38 knockout mouse, showing that in these mice, NAD levels actually were maintained during aging. And Eduardo Cini and his colleagues not only showed this in adipose tissue, but in multiple other organs, indicating that, at least in these organs, CD38 appears to be one of the major drivers of NAD decrease during aging. And so this really drew our attention, and we started working on this a number of years ago with Anthony Covarrubias, who is a starting postdoc in the lab. So the first thing that Anthony did, he had actually conducted his thesis working on adipose tissue, and was to go look in adipose tissue where Eduardo Cini and his colleagues had actually identified this process, this role of CD38, and asked, where is CD38 expressed in adipose tissue? And the way we did this was by preparing what's called a stromal vascular fraction. So we separate the adipocytes, which actually float when you centrifuge them because of their lower density, from a number of cells, the SVF fraction, which contains non-adipocyte cells. And we then further analyzed those for CD38 expression in mice of two different ages, the red and the blue mice, the blue mice being older. And we further focused our attention on lymphoid cells. And we can separate them, as you can see here, using a number of markers on non-leukocytes, non-macrophage leukocytes, and macrophages. And what was striking, it was that most of the CD38 expression in older animal was present in the macrophages. Now, we further characterize this by enumerating the total macrophages in adipose tissue, and this is shown here in a number of mice of different ages. And remarkably, we saw a progressive increase in the total number of macrophages during aging, from 2% to close to 6%. So this was already by itself an interesting observation. What was even further interesting is the fact that these increasing macrophages during aging are the so-called resident macrophages. The non-resident macrophages actually did not increase. Just to say a few words about these two different classes of macrophages that are present in most tissues. Many of your audience are probably familiar with microglia. These are the resident macrophages of the brain, or Kupffer cells, which are the resident macrophages of the liver, and so on. And it turns out that in most tissues, there are a number of macrophages that are deposited early in life, so-called resident, but there are also bone marrow-derived macrophages that can circulate and visit individual tissues. It turns out that during aging, the resident macrophages were the one increasing. What was particularly interesting is that CD38 increased, as shown on the lower left panel, almost exclusively in resident macrophages. You can see it started at around 1 and finished at around 4-fold increase in CD38 during aging. And non-resident macrophages did not show this process. Now, I want to stop to actually acknowledge the contribution of someone who had already discovered all of this in the 30s. And this is Elie Metchnikoff, who has been called the father of natural immunity. He, not only the father of natural immunity, probably the father of modern studies of the microbiome. Elie Metchnikoff is working at the Pasteur Institute in France. He's originally Russian, and is actually credited with the discovery of the macrophage, as shown here. And not only of the discovery of the macrophages of phagocytic cells, but also the realization that macrophages numbers increase during aging. And he was able to publish a number of papers for which he eventually got the Nobel Prize, and proposed a whole theory of aging based on the idea that the proliferation of macrophages during during aging was actually responsible for some of the manifestations of aging. I do not think that most of that theory, as he proposed, is correct. But however, he made some of the key observations that animate our work today and will continue to animate the work of many others. Macrophages are known to increase during aging and thought to play a critical role in the inflammation associated with aging. And this is illustrated here. 100 years later, we've learned a lot about macrophage biology. There are, in a very simplified manner, two broad classes of macrophages, so-called M1 macrophages, which are, as we know, increase during aging, generate a classical inflammatory response. These macrophages are activated, for example, by LPS, a bacterial product. There's another class of macrophages, so-called M2, which are mainly activated by interleukin-4. And these appear to be playing a role that is opposite to the M1 macrophages, a role of tissue and metabolic homeostasis, wound repair, and so on. We can actually isolate these macrophages and test them, and this is what we did. Anthony prepared M1, M2, and so-called M0 macrophages, and then looked at CD38 expression, and we saw that CD38 was exclusively expressed and at very high level in the M1 macrophages. And you can see here a time course of induction of the M1 versus M2 macrophages. Within 16 hours, we had close to a 500-fold induction of CD38 expression. And in agreement with this, if we look at an NADase assay, we can take a fluorescent substrate that is an NAD analog and measure the ability of the enzyme to cleave it. We can see that this activity is only present in M1 macrophages and is dependent on CD38. In red, I'm showing you macrophages that have been prepared from a mouse lacking CD38. You can see how they are unable to exhibit this NADase activity. So this raised a series of really interesting questions for us, is to understand what activates CD38 expression in these aging macrophages. And the first thing we did was to focus on a number of molecules that are called either pathogen-associated molecular patterns, PAMPs, or damage-associated molecular patterns, DAMs, which are different small molecules that are typical signature of either bacteria or viruses or of tissue damage. And macrophages have evolved a number of receptors that allow them to sense both the PAMs and the DAMs and to actually become activated and to clear the damage. And so when we did this, I'm showing you here the substrate that we used. We were struck by the fact that almost all PAMPs, the pathogen-associated molecular patterns, seem to be activating CD38 in these macrophages, and none of the DAMs actually did it in our hands. Now, the potential significance of this is the fact that we know during aging, there's a progressive disruption of the intestinal barrier. This is illustrated here. On the left, where we have an intact bacterial intestinal epithelium, and the bacteria actually restricted into the lumen. In contrast, during aging, we have a disruption where the paracellular translocation of bacterial product can occur into the lamina propria and actually lead to what I'm just showing you, that macrophage can become activated by these bacterial products and can increase CD38. So we think that this is potentially one of the mechanisms by which CD38 is expressed. And we will be testing this in the future by conducting experiments in aging mice in which there are no bacteria in their intestinal system. Now, another process that we have focused our attention on was the potential role of senescent cells. Now, we know that we've heard about senescent cells yesterday. We know they accumulate during aging in adipose tissue, and this is illustrated here on this figure, where using a beta-gal stain, which is somewhat selective for senescent cells, you can see if we compare a 12 months versus an 18 months old. You can see an increase in staining. This mouse has been treated with a senolytic drug. You can see a reversal to a low level of beta-gal staining. And this is present in two different forms of adipose tissue. Now, senescence itself is characterized by a tripartite phenotype of irreversible growth arrest, resistance to apoptosis, but particularly interesting is the so-called SASP, multifaceted secretory phenotype. So what is the SASP? So first, on this other side, I'm showing you the typical pathways that lead to cellular senescence. It can be either a stress, oxidative stress, or radiation, oncogene, pre-oncogene activation, or replicative senescence. And these senescent cells actually start secreting the SASP. The role of the SASP, which is a series of pro-inflammatory molecule, is to signal to other immune cells that will eventually come and eliminate the senescent cells. The SASP also has secondary effect, what we call paracrine effect, where it can affect neighboring cells, either by inducing secondary senescence or actually also induce promitogenic stimulation. And we suspected that the SASP might actually be activating CD38. And the way to test this experiment, again, conducted by Anthony Covarrubias. By the way, all of this work has been published in Nature Metabolism actually early this year. So there's a lot more than what I'm going to be able to show you during this presentation. So I invite you to go look at the paper if you are interested. So what Anthony did is to prepare supernatants from macrophages that had been actually, we started with fibroblasts, treated them either with doxorubicin or irradiated them. This is shown here on the left. This, we know, induces a very rapid process of senescence. We then harvested the supernatants and transferred them onto macrophages and then looked for the ability of these supernatants to activate CD38. And what Anthony was able to show is that both doxorubicin and irradiation-induced senescent cells supernatants were able to very strongly activate a CD38 expression in macrophages. And this was specific. We did not see this for other proteins such as that consume NAD, such as sirtuins or PARPs or CD157. Now, Anthony further showed this in using bone marrow-derived macrophage, BMDMs, and was able to show that we can actually also induce senescence in the cells themselves by irradiation. You can see them staining for a beta-gal here. And when we looked at CD38 expression, there was a very strong, again, more than tenfold induction in CD38 expression. Now, this led us to the following model that both the PAMPS associated with the so-called leaky gut and senescence via the SASP both lead to chronic inflammation. This has been shown before, but also lead to macrophage proliferation. One of the SASP protein is actually a macrophage proliferation factor. We're still working on this. It's obviously an interesting observation that could explain why macrophages increase during aging. We also observed, and I've showed you today, that this leads to an increase in CD38 expression, which leads itself to NAD depletion. Now, we believe that this depletion of NAD leads, again, as we started off, to metabolic dysfunction and sirtuin dysfunction. Now, this is quite interesting, but perhaps the most interesting aspect of all this is what can we do about this? And what I'm doing here, I'm showing you the graph that illustrated the competition between sirtuins and PARPs and the different ways in which we think we could be intervening in this process. The first way that the field actually has been working on is the idea of activating the sirtuins. Back in 2005, when the role of sirtuins in protection against aging became apparent, David Sinclair and colleagues started a company called Sir2 Is and actually discovered that resveratrol, a compound associated with wine, was able to activate sirtuins in vitro. And based on this, the company Sir2 was started. They generated a whole series of other sirtuin activating compounds, the so-called stacks, and documented in a number of elegant papers that the stacks indeed activated SIRT1 primarily. However, the stacks became somewhat of a disappointment in terms of their biological activity in humans. And one of the major reasons we believe is because they operate in the context of NAD depletion. So you can activate sirtuins, but if they don't have their main substrate, NAD, they do not work. So obviously, activating the sirtuin by themselves is not a solution unless one corrects the NAD levels. So another possibility that's been explored by Johan Hallmarks and colleagues is the idea of inhibiting PARPs. And indeed, inhibition of PARPs can increase lifespan in some lower organism, in a worm, but this cannot be done in longer-lived mammals, in which the PARPs actually play a critical role as well in DNA damage and repair. And so this is generally not considered a feasible approach for the future, although the PARP inhibitors are being considered as anti-cancer agents. Another approach that is being considered is to activate the rate-limiting enzyme in the salvage pathway. We talked about NAMPT being this key enzyme that sort of recycles the nicotinamide, which is shown here, generated by the PARPs in CD38 and CD157. It is rate-limiting step. There are a number of compounds have been identified that claim to activate NAMPT, and these compounds have very interesting biology and are being further explored for the potential role in reestablishing NAD levels. Another approach that's been taken is to actually bypass a potential defect in NAMPT and to restore NAD levels by providing precursors. And two of these precursors are being currently explored, actually being sold, nicotinamide riboside and nicotinamide mononucleotide, also called NR and NMN. And want to point, to go back to, finally, I'm going to discuss the supplement in a little more detail, but before this, I also want to highlight the fact that the final approach that people are considering as a potential treatment is inhibition of CD38, the activity of CD38. So the thinking is that one could not only replenish NAD levels, but if you do this in the context where there's accelerated degradation, for example, as I've shown you by CD38 activation, it is like filling up a sink in which there's a leak. So the idea of blocking CD38 activity is to block the leak and to allow the supplement to refill the pool of NAD. So what about NR and NMN? This is a figure that I showed you earlier, the salvage pathway, the kynurenine pathway, the price-handler pathway. Both NR and NMN are direct precursors to NAD. So if you look at the salvage pathway here, we go from nicotinamide to NMN, and then from NMN to NAD. Well, NMN can be actually purchased today. It can be taken as a supplement. There's evidence from the lab of Shin Ichiro Imai of a direct transporter that allows NMN to come into cells. There's another precursor, NR, which can itself, following the activation by a kinase, lead to NMN itself. Now, there's been a lot of debate as to whether NR or NMN are better as supplements. I think I will stay out of this debate. I think we should let the experiment speak for themselves. And I think there are a number of clinical trials that are currently exploring the potential role of both of these, either in comparison to each other or individually. And I think we will see. It could also be that these different supplements play a different role or will find different applications in the future. So I want to finish with what we can actually anticipate from these types of intervention. When we talk about strategies for boosting NAD levels, I'm bringing you here back to the left. we talked about NAD precursors, we talked about targeting NAD-consuming enzymes, but these are not the only intervention. It's known that exercise and dietary and calorie restriction by themselves actually enhance NAD levels. And this is where we got started. So it turns out that both diet and calorie restriction and fasting are all associated with an increase of NAD levels. So you can raise your NAD levels simply by these interventions. Obviously, the precursors bring this to a whole other level. Now, the expected effect of health benefits of these are multiple. As I mentioned, one that we would hope in the long term is a suppression of epigenetic drift by maintaining the epigenetic machinery in the best form possible. There's evidence in lower animal models for improved brain function and protection from neurodegeneration. There's, in terms of vascular system, increased neovascularization, increased capillary density and blood flow in different tissues. There's improved liver function, reduced liver steatosis, fatty liver, and increased capacity to regenerate. In the muscle, there's decreased atrophy, so hopefully an inhibition of sarcopenia, enhanced mitochondrial function, and increased physical activity. In the pancreas, there's evidence of improved beta cell function, increased insulin secretion, and reduced inflammation. In the adipose tissue, there's reduced dyslipidemia and a prevention of insulin resistance. And finally, there's evidence that some of these agents might play a role in reducing inflammation and improving immune cell function. So obviously, one of the key thing in the field right now is to make the transition between everything that has been shown in animal models, where modulating NAD levels can play a really important role in maintaining health, increasing health span, and the humans. And the field of supplement has run a little faster than the science. Many of these supplements, as I said, are currently available, and actually I suspect many in the audience are actually taking them. What will be really critical in the future is to conduct rigorous clinical trials that demonstrate both the safety but also the efficacy of these markers. I can tell you that many of the companies that I've been involved with or talking to are actually conducting these trials. So I think the next few years will be really interesting in this regard. So I want to finish by acknowledging many of the people who are participated to this work. I highlighted the work of Anthony Covarrubias, who was an academic postdoc. I also highlight the role of Rosalba Perrone in my lab, who has been working for a company that we co-founded, Napa Therapeutics, which aims to correct the age-associated NAD defect, but also Alessia Grosso, who is working on another collaboration, working on some of the NMN supplement. I want to highlight the collaboration with Judy Campisi, who has devoted her whole career to studying senescence. Birgit Schilling, who runs our proteomics lab and core at the Buck Institute, Arvind Ramanathan at the Buck, and Charlie Brenner at the University of Iowa, who now is actually in Los Angeles. And I will stop here, and I look forward to your questions.